And I think to introduce the last speaker, I think this is a nice progression here right before the break. As someone who's really helped shape this field and drive it as director of the National Institutes of Health Sciences and the National Toxicology Program, and has some great perspective on this, I'm sure. I'd like to introduce Dr. Linda Birnbaum.
Well, first of all, I think I'm going to very rapidly to try to go through kind of what NIEHS has been doing in terms of support of a lot of this work, and I feel completely inadequate because I'm a toxicologist. I don't really do much statistics. I don't do much big data. But what I do, which I heard from several people, is I go for help all the time. And I think that that's a very important thing. So what I want to do really quickly here is I want to just talk about kind of where we're all working in the data science landscape. And this kind of reflects the landscape where NIH is supporting lots of efforts here. In the high throughput technology, all kinds of stuff related to the different omics, the gigabyte scale digital imaging and many other techniques, and the computational techniques and infrastructures, things like cloud computing and high performance computing. And then we absolutely must be sure that the next generation of data science relies on being fair. There's a term I'll use: repeatable, findable, actionable, interoperable, and reusable. And all I can say to everyone is, if you think you're going to be able to keep your data yourself, that's not going to happen. Data is going to be shared. We already know that is being required in the genomics range. It is also going to be required in the exposure science. There are a lot of ethical issues that have to be worked through here. You know, your genomic data is your own. Your exposure data may be what somebody is doing to you. So there are issues, but we've got to really acknowledge that and go forward. So I just wanted to very briefly talk about all the work that we've done, really since the emergence of our last strategic plan in 2012, where actually one of the key platforms of that plan, goal 3, well, one of the overarching themes was exposure science. And then when you get to our goal 3 was really the development of the exposome. And I think the whole issue of knowledge management and data management has been something that has played an absolutely major role in where we've been for the last couple of years. We've held a series of workshops, and I probably should have had these listed chronologically, but I've kind of have certain things. Overall list is chronological, but we've held a series of workshops. So we first, 6 years ago, we held a data sharing workshop for environmental health sciences. We then held a workshop in identifying opportunities for global integration of toxicogenomics data. In 2013, we actually had a data challenge that we did with the National Center for Advancing Translational Science and with UNC and with Sage Biologics. And this was a toxicogenomic challenge, which I think was extremely important. And the objective of the challenge was to obtain a greater understanding about how a person's individual genetics can influence both cytotoxic response and genomic data as well, to develop that integration. In 2013, we sponsored the first of the National Academy meetings through the EHESD Committee for Integrating Environmental Health Data to Advance Discovery. And then in 2013, remember, we're part of the big NIH, even though we're located in the southern part of heaven and not here. But we dealt with the issues of community-based data and metadata standards. And under the Common Fund, NIH put out a huge call to fund many centers, especially focused on training, to begin to develop and support people in the data science arena. In 2014, we held a workshop which was looking at a framework of environmental health science language. You got to be sure that everybody is speaking the same language. We heard from Chris about DREAM and about CHEER, and I'm going to come back to CHEER in a minute. But the point is you've got to be sure you mean the same thing. So the development of ontology is really an important concept to go forward with. And then we actually are the Office of Health and Assessment and Translation, headed by Chris Thayer for many years until she deserted us. Sorry, Chris, for IRIS at EPA. But anyway, LED basically does literature-based environmental health science assessments where you're really trying to integrate all the information and make it fully transparent. Remember, that is something that is absolutely essential. And then in 2016, we had a second ESEHD workshop where we were looking at the best practices for sharing data from environmental health research. So I'm going to very briefly talk about what's been going on at NIEHS, both intramural and extramurally, in terms of the issues of data, of really big data and data science. So we established a number of separate but totally matrixed together offices. First is the Office of Data Science, which Stephanie Holmgren is the acting director of, and that is providing leadership and the support to enable discovery, access, and use of data to support and advance research policy and decision making. And it's really focusing on data integration and metadata management. And I should mention, and Stephanie, I know, will not feel bad when I say this because we are currently recruiting. We just finally, after over a year of controlled hiring, and that is being polite, we have just been given the go-ahead to begin to recruit for this position. So anybody who's interested, please let me know. Anyhow. We also had established 2 years ago an Office of Scientific Computing, which is focusing on the issue of cloud computing and high-performance computing. And we have the expertise and staff and resources that is headquartered. I should mention that the Office of Data Science, because of its extensive use of many large data management tools and so on, is headquartered in the division of the NTP, and the Office of Scientific Computing is headquartered in the Division of Intramural Research. We will soon be recruiting, I've gotten the go-ahead to do this, for the Director of Environmental Health Sciences Cyber Infrastructure. And what this is, we've kind of set up a matrix accountability where that person will report directly to me in the Office of the Director, but will maintain tendrils and integration between the data science, the high-performance computing, the web information, and all the social media kind of stuff, and the basic enterprise computing to make sure that we've got the capability to, the hardware, the software that we need to do the work. We've also begun other hires, so we're interested in hiring a data scientist and a developer, and we recently hired a data scientist in our extramural program. And when I've been talking about ODS and OSC, those are also involved with things that many of you in the extramural community and our grantees, you'll all be hearing about some of the work going on there and the opportunities. And we're providing a lot of training, especially in-house, to all of our scientists related to machine learning, database structure, and different kinds of programming. Now, you've heard about CHEER, so I don't have to go into any details about it, but I think the importance about CHEER is that it is this integrated laboratory network where you have a central system. That's actually where you say you want to send your data, and they control all the data flow and so on. And then you have 6 laboratories in different parts of the country that have some different expertise. Some are doing the agnostic analysis. Some are doing more targeted analysis. Well, they're all doing targeted analysis. Some are looking at some early response markers. But as Chris said, there's got to be an integration between them so that there's a lot of QA, QC going on, led by the group at Mount Sinai. We are very excited about this. And I should mention that this was funded by the end of the National Children's Study, the money that was available that year, so it was forward funded. It is funded through this year. We are looking forward for what will happen as we go forward. There is some funding for it from the ECHO program, which is kind of the successor to the National Children's. But I would urge you to talk to people about how important this is, not only for children's health, but for everybody's health. And so we are looking to morph this into here, where we take off the C, and it'll just be a health environmental assessment, resource for across NIH that will be able to take samples from anyone who has had NIH-funded work. We are busy talking to the All of Us, which is the the new name for the 1 million person NIH precision medicine cohort, and their interest in being able to utilize this resource. Frankly, it's such a great thing. All right, I'm very proud of what we've been able to set up. I want to see it continued, and I would like to see even the opportunity potentially to enrich it with a couple of additional laboratories that have the capability. But when we look at this data repository, at what we are providing here, it will allow us to have the clinical, biological, and epidemiological measurements are all being put into the databases. We're going to be able to look eventually at familial relationships. In many of the children's studies, we have mother-children information. And in some of them, we have sibling linkages we can do, and we can look at multiple time periods across fetal and child development. So what came up here is really just the, this is the kind of measurement data that we would get, for example, for looking at the phthalates and all the different phthalates and all the different metabolites that are being monitored there. So I should have said SHEAR, of course, is an extramural effort. Unfortunately, so far, those of us at NIH can't access it for our own specimens unless you have an extramural collaborator, and then you can. But the comparative toxicogenomics space, which was established by Carolyn Mattingly at North Carolina State, is actually a real opportunity here for us to look at all the interactions that toxicogenomic connections. And if you look kind of at the map here, you're looking at gene ontology and the chemicals and the different genes. But now we're beginning to bring in the whole exposure piece. So we have both stressors and receptors, the biomarkers that we might actually be able to measure to help inform us about the chemicals, and some of the outcomes, which will be important here. So this is another resource that we have established that is available for access and use. In NTP, we have a lot of toxico-informatics, as we're calling it, and lots of different platforms and databases that are being developed. So, for example, we've got the benchmark dose express 2, where you can recalculation for risk assessment, lots of utility of the TOX21 data. TOX21 is the coordinated EPA, NCATS, NIEHS, and now FDA platform looking at thousands of compounds. We've got Drug Matrix, which involves about 600 different pharmaceuticals that have been, we have the data for them, where you have both phenotype as well as the exposure information and and results with a correlation browser. The Integrated Chemical Environment is a program that's looking at the data from all the alternative systems that NICEDM is working on with the different ICVAM partners, and the Chemical Effects and Biological Systems, better known as SEBS, where initially this is where NTP has put all of its testing data, both the short-term and the long-term data, but we are also now having more and more data put in it from grantees. So, for example, the large BPA Clarity study, which is involving the FDA 2-year guideline-based study, an expanded guideline-based study, and then all the grantees who got either whole animals or pieces of the animals that they could look at a number of tests. All that data has been entered into the SEB system, and this is all freely available. So again, what we're trying to do is really look at integrating the chemical gene expression path, all the different kinds of data, and make it fully available. So I've talked a little about what we have at the Institute. I want to remember or just remind you that this is all captured and included in what the big NIH is doing, which is the development of the whole data commons kind of approach, where we have co-location of the large and or highly utilized, I don't know why I keep looking up here. Anyway, I got it down here, of large and highly utilized, the NIH-funded data with storage and computing infrastructure, using a lot of tools for analyzing and sharing digital objects. And of course, not to be outdone, NIEHS is establishing its own data commons to integrate the NIEHS data, make sure that it can integrate with the larger NIH data as well. So we're serving on lots, we're involved very much with the whole NIH program, and we're working with a team at UNC to make sure that we can move our data and metadata between NIHS and NIH. This is not a trivial kind of issue. So we're also trying to really increase the whole interoperability of the NIHS data systems. We are trying to be sure that all of our different, I mentioned a couple of these different platforms we have, the SEBS and the ICE, which are the NTP kind of efforts. We are establishing our own data commons related to all the intramural research programs with Drug Matrix I mentioned, and that EPR and REDCap are two clinical large data sets that we have. The Environmental Polymorphism Registry was started about 15 years ago. We have collected both biospecimens from almost 20,000 people in the North Carolina area. And it was an opportunity when we wrote the consent forms at that point. We said, can we contact you again? And the answer was yes from these people, and we've been able to recontact, and we're now actually conducting a number of studies that are actually really kind of exciting, where we're looking at the relationships between the exposure of these people, as we can measure largely by questionnaire data at this point, and with their genotypes as well. And REDCap is another clinical database that we're looking at, and trying to we have all these assets. We're looking at that the programming interfaces. Eventually, we want to get back to the end users through a lot of software applications. So the road ahead is towards the information commons and knowledge network. And this is really a huge challenge for computer and information sciences. Here, the key that we're dealing with here is that an information commons provides broadly available biomedical knowledge on large populations of patients for research, while knowledge networks provide additional value by integrating the data and evolving knowledge of fundamental biological processes. That knowledge can then foster disease classification, new clinical approaches, basic biological discoveries, and disease prevention. And by the way, if anybody wants these slides, all they have to do is send me an email and I'll forward them to you. We really have found that a breadth of expertise is necessary, and we've heard this before, the need for multi and transdisciplinary work here. We need both clinical, biological, environmental, computer science, information science, and we also need translators, individuals that can kind of straddle multiple worlds and build bridges. Now, that was the big environmental information commons. We also need one for environmental health as well. And this is what we're kind of doing internally. And I briefly mentioned the EPR program that we're looking at right now, where we are calling back people with certain genotypes that we've identified who we think, in fact, might be asthma sufferers. And we're trying to identify, are there subtypes of asthma that have different genetic susceptibilities that can inform what kind of treatments will work best for these sufferers. And again, we'd have populations of scientific interest. So we're working very closely. This is an intramural program, working closely with our colleagues at UNC. And we have data on air quality and socioeconomic. We have extensive patient surveys, both on their health and on their potential exposure. As I said, we have their genetic and we have biomarker data. And then we're also looking for the other kind of aspect, not starting from here's a person who has a disease or here's their genes. We're starting from the point of, well, you know, here's a certain chemical. Can we determine what the properties are of that chemical to guide our subsequent testing? So these kinds of use cases that we're going through right now, we're hoping will drive environmental health science and the building of data integration technologies and models for collaborative science. So I just kind of briefly want to, you know, talk about the workshop charge. I think Kim said a little at the very beginning, but the issue is here we really want to look at the promise and the potential pitfalls of environmental health data integration. And we really want y'all to work together to conceptualize where do we need to go in future research direction. I really think this is an opportunity for all of us, and I say that because I have a day and a half where I can stay and think science and learn before I go up to Bethesda. Anyway. but really that we can think about and discuss the specific research questions that can guide those who are building the information commons and the knowledge networks, whether for environmental health or for other purposes. And I just wanted to urge you, we have a new draft strategic plan, the 2012 to 2017, which I think was pretty exciting. We have a new draft out for your comments. Please that's the website, the link for it. We're looking for feedback from the community. It will be open for feedback until March 30th. We did take this to council just a week ago and got their comments, and so that was great. And I do want to thank explicitly Kimberly Thigpen-Tart for helping in developing this presentation for me, because I said I'm really here to learn. So thank you all.
Any questions while we have Dr. Birnbaum up here?
Okay. Is this on now? Yeah. My name's Joan Aaron, and I'm a consultant, and I'm very much interested in the FAIR principles and the data sharing and all of where this is going. And I'd like your comment on how do you see this playing out with the new federal requirements for federally funded research for when something is published, that the data be made available? You know, I see all this complexity, and if you could comment on sort of what's happening there, what is happening there, and perhaps what should be happening there.
It's a sticky wicket. As I said when I talked about this, that any federally funded data is going to have to be made available. You know, you can't hide it. You can't keep it to yourself. We are busy, we and many other people are busy working on how we can do that in a fair manner. And by there, I'm not talking about the the term fair, but in a way, there are ethical issues that have to be dealt with. We know that all genomic data, for example, that NIH funds has to be put into dbGaP. And I think we are just beginning to work through the issues of what do we do with federally funded exposure data. And I said it's really a much more, I think, difficult situation about how do you provide anonymity to some exposure data that somebody might have done to you. I actually served on or chaired an SAB for a long time looking at a big exposure study. And what was interesting there is that if, you said this is a 65-year-old man and he lives in this area along the river and he has a heart condition and COPD, and it's a small population, guess what? You know who that person is. So these are huge issues that we're going to have to deal with. But I don't have any answers for you yet.
Okay.
But thank you for asking.
If you wouldn't mind it, you can turn on the mic.
Oh.
Any other questions?
Oh, did I?
No?
Thank you. Great, thank you.